The last video talked about transcription, going from DNA to messenger RNA, and now we're taking that message that left the nucleus, we are in the cytoplasm, and the ribosome is going to make it into a protein, specifically a polypeptide chain. So a little review is we need to go from DNA to mRNA. Why? Because DNA is too big and we have to protect it. Can't risk having it moved around getting damaged. So we are going from the nucleus to the cytoplasm. And the cytoplasm is the site of where translation will occur. Whereas the nucleus is the site of transcription. So, in regards to making a protein, we are specifically going from mRNA to our protein. That is translation in a nutshell. So think of it as if you are translating languages. It's almost like we are going from a recipe to food. So going from one thing to another thing. And we use ribosomes to help make this happen. They read the sequence and then assemble the amino acid sequence, the polypeptide chain. Everything stems back to DNA. So think of mRNA as a one particular recipe and DNA as the overall cookbook. So how to read RNA? 3-letter sequences called a codon. One codon per amino acid. So this codon will be one amino acid. This codon will be translated to another amino acid. And as you have the amino acids that are bonded together, that is going to make your protein. Now your codons are specifically going to code for your amino acids. So we use what's called our codon chart to figure out how to do that. For example, if you had the code GUA, we would look at the chart, notice how it's split into 4 different squares. We would see the square that has the big G. We would then go down a level, the medium-sized U, and then we see 4 different letters right here. G, A, C, and U. This is telling us that the amino acid valine has 4 different possible codes that will code for it. In this case, we just want GUA, but it also has GUG, GUC, as well as GUU, all code for the amino acid valine. Another example of the table. Kind of figure out how to navigate through. Just another look, same general idea. Now, whenever we have our amino acid sequences, we always have a start codon. The sequence cannot start until you locate AUG. And the amino acid that this codes for is methionine. If you don't have AUG, then you don't have a sequence. You need to start with AUG every time. And then at the end of the sequence, you will have a stop codon. And we have 3 possibilities for a stop codon. UAA, UAG, and UGA. So you need a start, and then you'll have some codon, and then you'll have some amino acids, and then you'll have a stop. Now, the stop codon is not an amino acid. It simply is just basically a symbol that the sequence is over. So in practicing kind of reading, trying to figure out mRNA to amino acid. So AUG. Let's do a couple different colors here. So we have A, U, G, methionine. And that's what we see, methionine. That is our start codon. Every single time AUG is our beginning. GUC. G, U, C, and we have valine. AGA. A, G, A. Looks like arginine. CCC. C, 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 proline. And then finally UGA. UGA, and that is one of our stop codons, which tells us that this sequence is over. Now the last thing is these are all going to be bonded. And each of these bonds is going to be what's called a peptide bond. That's what bonds amino acids together. So one sequence, it looks like we got 1, 2, 3, 4 amino acids before we hit the stop codon. So, kind of how does this work from a process standpoint? Well, when the RNA is in the cytoplasm, basically a ribosome will attach to it. And remember, the ribosome has the rRNA, ribosomal RNA. When the ribosome attaches to it, it basically signals to transfer RNA to come over and help deposit the amino acids for translation. Your transfer RNA will bond to the mRNA with their own codon. This is called an anticodon. When this happens, it is basically making sure that it has the correct amino acid to drop off. So it's almost like a system of checks and balances. If the anticodon complementary bonds with the mRNA codon, then it will drop off the correct amino acid. And then when it'll leave the ribosome and then go to try to find another amino acid. So this will continue as the ribosome moves down the line. Different transfer RNAs come and kind of basically drop off the correct amino acid. When they reach the stop codon, it basically signals that the sequence is over. And then we end up having a string of amino acids, our polypeptide chain. So think of like your ribosome as the builder, and basically the transfer RNA kind of being the helpers to bring the necessary tools to finish the job. So transfer RNA, they carry the amino acids. Remember like we just talked about, the anticodon. This bonds with the codon to simply make sure that the correct amino acid is being brought. Each transfer RNA is going to be specific to that amino acid. So you're going to have essentially 64 different transfer RNAs because there's 64 different combinations. So this one would code for U in terms of mRNA. U bonds with A bonds with U, C bonds with G, and this is going to bring methionine because AUG is our start codon. So that's signifying the start of the sequence. kind of another look at kind of what's happening. Kind of moving down the line, it'll drop off and then leave, and then the next one will come in to deal with the next codon, and then as they have multiple amino acids, these will be bonded together as the ribosome moves down the sequence of messenger RNA. So as we see now, we have two different transfer RNAs. Methionine will bond to the next one, methionine and then leucine. So we see kind of the sequence starting to bond. And then as the ribosome moves down the line, new transfer RNAs come and then continues that trend. So the growing polypeptide chain, as the ribosome moves down the line, the leucine will then bond to the alanine right there. And what we end up having is a polypeptide chain with multiple amino acids, a start and a stop. It's stopped at the stop codon here, UGA. And then this could be thousands of amino acids long. And what will happen is as it gets big enough, it'll start to bend, and it'll start to fold, and the more complex it gets, the more folds it has. But that'll be for another day to talk about protein structure. So, overview. DNA replication. This is specifically for cell division. The whole point of this is DNA to make DNA. One DNA makes two DNA molecules in preparation for cell division, where one cell becomes two. This happens in the nucleus because that is where DNA is. Transcription. This also happens in the nucleus because that is where DNA is. What we're focusing on here is DNA making RNA. Why? Because we need to get message out of the nucleus. And then when we are out of the nucleus and into the cytoplasm, we are going to do translation. Translation, we have RNA, which came from transcription, and that is going to make protein. This is happening in the cytoplasm, and this is happening with the help of your ribosome and your tRNAs. So another quick practice. We're going to go transcription and then translation. So DNA to RNA. Well, TAC, remember DNA has T's. RNA, remember A bonds to U. Where in DNA, A bonds to T. So what we have here, TAC, is going to be AUG. CGG, GCC. TAA, AUU, CGT, GCA, ACT, UGA. Remember, these are complementary. So we have to follow the base pair rules. Now we are going to go similar to what we did before. Go from RNA, messenger RNA, to protein. We have to use our codon chart to assist. AUG is our start. You should become familiar that this one is always going to be methionine, which is our start. The This is still an amino acid though. GCC, we have GCC, that's alanine. Then we have AUU, so AUU, and it looks like that one is isoleucine. Next one, we have GCA, so here's GCA. Looks like we have another codon that codes for alanine. And then finally, UGA, and this should be one we get familiar with because there's only 3 stop codons. So that tells us that the sequence is over. And remember, these are bonded together, and that bond is called a peptide bond. So going from DNA to RNA to get out of the nucleus, RNA to protein to make sure we code the message. Now real quick, one other difference that we see up here. DNA to make DNA, this is going to be the whole molecule, the entire double helix. And this is only going to be a segment. We're not transcribing the entire thing, and that segment is called a gene, a sequence that will code for a protein. Now, final 2 things here. When we look at our amino acid, remember your amino acid is your monomer for a protein. The basic structure of an amino acid. The only thing that changes in the structure is what's called this R group. And we have 20 different R groups, which means that we have 20 different amino acids. So if we look at that codon chart, we're only going to see 20 different names. Stops are not amino acids. So 20 different names. And whenever you have that chain, we call it polypeptide. Poly means many. So many peptides, think many peptide bonds, amino acids, or amino acid chains. Final thing, always remember our start codon is AUG, which is an amino acid called methionine. And then we have 3 stop codons. These do not code for anything. They simply signal that the sequence is finished. So you need to find AUG. That's your beginning. You need to end with one of our 3. If you don't, then you don't have a finished sequence. All right, that is it for translation and protein synthesis in general. If you have any questions, please ask. Thanks for watching.